The overall goal of this procedure is to set up a culture system that closely recapitulates the cellular and intracellular microenvironment of human bone marrow. First, the surface of a tissue culture well is coated with the endosteal matrix. Then, bone marrow mononuclear cells or other cells of interest are mixed with the bone marrow matrix. Later, the endosteal matrix is removed and the cell matrix mixture is added to the well. Once the matrix has gelled, it is overlaid with growth medium. Ultimately, cells can be visualized directly in the matrix or can be isolated for downstream analyses.
Compared to existing methods for culturing cells, for example standard tissue culture methods or the co-culture models where cells are grown on plastic, the main benefit of using a 3D culture method is that cells are grown within the physiological microenvironment, and hence it allows you to study cells within the native conditions of the tissue.
Prepare a 48-well plate in which the cells will be plated by first adding 65 microliters of endosteal coating solution into each well. Spread the solution evenly to cover the entire surface of the well. This method is scalable to other surfaces as well. Allow the plates to incubate at room temperature for at least 30 minutes. In the meantime, prepare the cell suspension of 50,000 cells per microliter.